Hello, my name is Sade Cromratie Clemons. I'm a master's student in the geography department at CU Boulder, and I study tree ring response to flooding in terms of carbon storage and transport in the Colorado Front Range. Specifically, I want to figure out where large wood pieces that have been transported by floodwaters come from by looking at the tree ring measurements and oxygen isotopes. Large wood is important in many aspects, from sediment retention, channel morphology, and habitat creation in river systems, but as stated before, large wood is also important for carbon storage and transport because trees recruited to streams are important carbon stock. Being able to figure out the origin of wood in and around a channel will provide more information on how to quantify and track contributions of large wood from different sources to the overall carbon budget. For this study, my samples were taken from West Creek, a stream that starts in the Rocky Mountain National Park area and drains into the North Fork Big Thompson River. In the picture, you can see me and my advisor standing in front of a massive log jam, and this is where I sampled from. My samples contain both subalpine and montane tree species, specifically ponderosa pine, Engelmann spruce, and Douglas fir. Samples were taken from log jams in the West Creek stream and around the West Creek stream, and from standing living trees at different elevations next to the stream. Once I collected all my core samples, I cut and ground each individual tree ring. Each ring constitutes 1 year of tree growth, and I need the rings to be split up because that's how I will compare the logjam and standing tree samples. Because I cut the rings by year, each year now represents one individual sample. This summer, I focused on prepping and processing my samples. The samples have been prepped in the lab by bleaching and extracting all the extra oils and resins from the wood. The result of this process is cellulose. Cellulose can be found in trees and provide support to the cell walls. It can also be used to find oxygen values of each year. Instead of using all of the wood, I use cellulose because wood has components like lignin and impurities that can alter the oxygen isotope value. With cellulose, I can feel a little more confident that my values are not changing because of other components reacting in the wood. Using the cellulose extracted from each tree ring, oxygen isotope analysis is being run using a thermal conversion elemental analyzer, which can be used to calculate isotope ratios. For my study, I am using it to calculate oxygen in terms of delta 18O, which is the abundance of carbon 18 to carbon 16. The instrument breaks down the sample into hydrogen and carbon monoxide. These gases are put through an isotope ratio mass spectrometer that measures the abundance of oxygen and hydrogen in a sample. I'm interested only in the oxygen values calculated from this analysis. I use oxygen isotope values because they are a really neat indicator of climate conditions. When trees take in water, the oxygen isotope value from water is recorded in the tree ring. The oxygen value of one ring can tell you what the growing conditions were like for that tree during that year. For example, trees growing in drought conditions at higher altitudes will have a higher carbon 18 value because the water taken up by the tree has a higher carbon 18 value. Lighter isotopes evaporate first, so in this case, carbon 16 would evaporate out of a water source first, leaving more of the heavier isotope, carbon 18, behind. The evaporation rate is also higher at higher altitudes. Trees in these conditions will have different oxygen isotope values than trees in less evaporative areas at low altitudes. Because conditions are drier near the top of hills versus at the bottom, and there is a change in altitude, I'm using the oxygen isotopes to compare isotope values of standing trees at varying slope positions to those of the large wood pieces based on that climate trend. Based on what the isotope values of the tree rings say, I can use that data to try and figure out the origin of the large wood in the stream. My samples are in the process of being analyzed, and it will be very interesting to see the results. From there, I can calculate carbon using published estimates and figure out the contribution of the hill slope and riparian trees to the carbon in the logjam. There was a lot of lab work that needed to be done this summer. I would like to thank the Indian Peaks Wilderness Alliance for being an integral part of my lab work, especially the isotope analysis of my samples.